Assalamu alaikum. My name is Dr. Saba Zafar, and I am doing my job as an assistant professor in the Department of Biochemistry and Biotechnology, the Women University, Multan. Today, I am presenting a topic, keratosis obturans, a study of complex hearing loss condition in mouse model. This research is carried out at the Department of Craniofacial and Developmental Biology, King's College London, UK. So what is the keratosis of Terrans? It is an external canal disease characterized by the accumulation of dense plaques of disseminated keratin in the ear canal. Keratin in the ear canal. It causes acute hearing loss and severe pain. Its pathogenesis and etiology is not clear, but it is observed that it is caused due to the chronic hyperemia and accumulation of epidermal keratin debris due to irregular movement of epithelial cells resulting in keratosis obturans and cause hearing loss. If you can see here, this is the picture of an human ear, and it is the external ear canal, which is filled with the keratin, which is the phenotype of the keratosis obturans. So to look into the research, we have an idea. We have to an idea about the wind signaling pathway. In this figure, you can see the two conditions when the wind signaling is off and when the wind signaling is on. So what is the wind signaling pathway? It is involved in the proliferation and development of the cells. Wind signaling pathway is of two types. One is the calcium dependent wind signaling pathway, and other is the beta catenin dependent wind signaling pathway. In this research, the wind signaling pathway is involved is beta catenin dependent because beta catenin is an product of the wind signaling pathway, which is involved in the proliferation of the cells. So here you can see the pictures of the cells. There is an a beta-catenin stabilizing complex inside the cytoplasm of the cell, which composed of the different proteins. And you can see here that this protein complex phosphorylates the beta-catenin, and it is followed by the degradation of the beta-catenin when the wind signaling is off. When you can see here, when the wind is on, this beta-catenin stabilization complex is combined with these cell membrane receptors, which are the frizzled protein and LRP6. And there is the no phosphorylation of the beta-catenin protein, and there is the production of the beta-catenin, which cause the proliferation of cells. It is observed that where there is a more beta-catenin, there is a more proliferation of the cells. And we will study the function of the beta-catenin or wind signaling in the keratosis obturans because there is also the hyperproliferation of the epithelium cells. So move further. There I want to mention this GSK3 protein because we studied this GSK3 protein in the mice model. We knocked out the exon 3 in this protein and study what happened with the beta-catenin. What is the activity of wind signaling in the air? There is a little research about the function of the wind signaling pathway during the ear growth and disease, but it is observed that is then gene ethol1 transcription factor, which is involved in the differentiation of the differentiation of the hair cells in the ear. So what are our study goals? We want to reveal the underlying pathology and development mechanism of keratosis obturans, address the location of FSP1 positive fibroblasts using FSP1 YFP yellow fluorescent protein transgenic mice. What is the FSP1? FSP1 is the protein which is also involved in the proliferation of the cell. So to determine the effect of the wind signaling activation in the FSP1 positive fibroblast on the external canal epithelium and its role in the pathogenesis of the keratosis obturans. So what are the material methods? We use two types of transgenic mice in this study. One is the FSP1 free negative beta-catenin flox exon 3 wild type mice. Second one is the FSP1 free positive beta-catenin flox exon 3 mutant mice. Third is the FSP1 free yellow fluorescent protein 5-week-old reporter mice. And last one is the Exin 2 lakh Z4-week-old wild type mice. So we did the histology and antibody staining. We did the tissue processing and sectioning, cryo-sectioning of the tissues, trichome staining, immunohistochemistry, and the X-con staining. So we used to make our knockout mice model. You use the Cree-Lox P system. What is the Cree-Lox P system? There is an overview of the Cree-Lox P system. In this system, the Cree recombinase enzyme is used. This enzyme removes the sequence between the two palindromic sequence of the LOX P site. If you can see there, from there to there, what is the one palindromic sequence, and this is the second palindromic sequence, and these sites are called the LOX P sites. There is a 8 base pair sequence there. So this sequence is used in the recombination system in the transgenic mice. If free is present in under the tissue-specific free driver or in promoter, when the promoter activates, it activates the free recombinase enzyme produced, which removes the specific gene between the two LOXP sites, and the two LOXP sites recombine, so inactivated the gene in the specific tissue. So in our research, we use the FSP1 Cree beta catenin flox exon 3 mutant mice pre-LOXP system. This is the FSP1 Cree mice in which there is a FSP1 promoter which is present before the free gene. So when and there is another mice that is the beta catenin LOXP mouse in which two LOXP site, and in which way there is the beta catenin exon 3 present. So when these two mice crossed, there is the mice for FSB1 Cree positive beta catenin phosphorus exon 3. In this there is a mice the beta catenin exon 3 removed, which is present in the GSK3 protein, which is present in the GSK3 protein. And if the GSK3 protein did not phosphorylate the beta catenin, there is the overproduction of the beta catenin. Second is the we're using the same technology and made a FSP1 Cree EUIFP mouse in which in between the two LOXP side there is the transcription blocker. When this mice is made with the Cree mice, and this Cree removes the transcription blocker, and there is a production of the yellow fluorescent protein, which indicates that there is the FSP1 cells are present. So what are the results? Identification of phenotype, if you can see here, there is the mice, this is the human ear and this is the mice ear, and it looks similar as the mice ear canal is also blocked due to the keratin. Here I mentioned that this mouse model is originally generated in order to study the wind signaling in the cancer studies, but one defect in the ear canal mimicking the human condition, keratosis obturans, we use this mice to study the condition. So if you can see here, there is the identification of structural changes by trichome staining. We did the trichome staining on the tissue sections from the 4-week-old and 8-week-old wild type and mutant mice. And if you can see here, the ear canal of the wild type mice is normal, while the mutant mice is filled with the keratin. Second, if you can see here, there is a remarkable difference in the epithelium and cartilage of the wild type and mutant mice. This is the epithelium of the mutant mice, and this is the cartilage of the mutant mice, which is the more proliferated, and cartilage is more ossified in the mutant mice in comparison to the wild type mice. Third, we used to study the EYFP mice to see that where the FSP1 protein is expressed in the ear canal cell. So if you can see here, the FSP1 protein is expressed in the cartilage and also in the fibroblast beneath the epithelium. But you can see there is no FSP protein present in the epithelium. So what is going on the epithelium? Why the epithelium is proliferated in the FSP1 mice, and why there is the overproduction of the beta catenin? To confirm the presence of the beta catenin, we also use the reporter mite of axin 2 lag Z reporter mice. So where there is axin, there is the production of the beta catenin. So you can see that the axin is present in the epithelium as the blue staining and also in the cartilage. Then we use the beta catenin staining to observe that where is the production of the beta catenin, and in this you can see that there is in the mutant mice there is the production of uh, beta catenin or beta catenin signaling present in the epithelium and also in the cartilage. In comparison to the wild type mice, there is the overproduction of the beta catenin. To differentiate between the pattern of differentiation, we use the suprabasal and basal markers K10 and K14 proliferation markers. And there are the remarkable difference as you observed in the picture. There is the more K14 and K10 cell proliferation in the mutant as compared to the wild type cell. Then we use the PCNA staining to count the cells in the mutant and wild type, and by the statistical t-test, it is observed that there are more proliferation of the cells in the mutant than the wild type. Then it is confirmed that the beta catenin is present in the epithelium and in the cartilage also. There is a more proliferation of these cells, but we also observed in the EYFP staining that FSP1 is not present in the epithelium. So how the beta catenin goes into the epithelium and what happens to the epithelium, we perform that topic, we perform the staining for the EGFR receptor, and we observed that there is the more EGFR staining in the epithelium of the mutant mice, which shows that something is signaling from the epidermis to the dermis. So what is the conclusion of our research? There is an overexpression of Wnt signaling in a subset of dermal fibroblasts lead to significant changes in the overlying epithelium, driving alteration in proliferation and stratification. Some cases of KO might be due to defect in the dermal fibroblast rather than being a problem with the dermis itself. So there are the future perspectives that what are the FSP1 Cree labels in the mesenchyme, which signaling is present between the epithelium and mesenchyme, and applying a therapeutic in the ear at early postnatal stages. Thank you for your listening. Have a good day. Allah Hafiz.